Hi, could somebody confirm you can hear me?
Yes.
Yes.
Thank you. I was actually hoping to take my computer in today and get it worked on to get the time correct, but it'll have to be tomorrow. All right. Are there any questions before we begin? All right, let me share my screen. That's what I want. Quick. Here we go. All right, you should be able to see my screen now. Today is April 13th, and we should be on chapter 2. We have not started chapter 2 yet, but we will start it today. I'm pretty sure we finished chapter 1. Is that correct?
Yes.
Thank you. Chapter 2 is on chemistry, and I can briefly state that there was talk in the class and the microbiology lab instructors about removing chapter 2 and then putting in another chapter later on. But now I'm opposed to that because students tend to be kind of weak on chemistry, and chapter 2 is chemistry. And I think the review will be good for you. I don't have any other glasses here, so you're going to have to live with these glasses. Oh, there they are. So the normal slide I give you is an introductory slide giving you the major goals and a rough outline. Know the terms of chapter 2. Excuse me, I've got a frog in my throat. What's an organic and an inorganic compounds? What are hydrocarbons? What are polar and nonpolar molecules? What are hydrogen bonds? What's an acid, a base? And what is the pH? And then with respect to the 4 biological molecules, be able to describe the relationships and the characteristics of the monomers and the polymers, be able to describe the composition, the structure, and the function of the 4 major biological molecules, and then be able to give examples of each. Any questions about what we're going to do? Let's see if I can raise that. All right, so this is showing you the scale of things, a picture from your book. You should know the relative order of things in this chart. Let me shrink that down a bit. Why can't I get that all in there? There we go. So you should know that atoms are the smallest thing and small molecules are bigger than atoms. Large molecules are bigger than small molecules, and a large molecule would be something like a protein. You should know that viruses are bigger than proteins, and that prokaryotic cells, like bacteria, are bigger than viruses. Eukaryotic cells are bigger than bacteria or prokaryotic cells. And then we have frog egg, which is about the smallest thing you can see. with your eyes, bigger than a eukaryotic cell. And I should mention that a frog egg is a eukaryotic cell. It's just one of the bigger ones. And then the biggest eukaryotic cell is actually that you've seen, I should word it that way. The biggest one that you've seen is the yolk of a chicken egg. Okay. Now, if you've seen the yolk of a turkey egg, and I once had a student who said that they had, that's even bigger. And if you've ever seen the yolk of an ostrich egg, that would be the biggest one possible, at least at this point in time on the planet Earth. However, not many of you have ever seen probably even an ostrich egg, and I've certainly never seen the yolk of one. I've never even seen the turkey egg yolk. And then a person, of course, is bigger than a chicken egg. So you should just know the relative rank. You don't need to know that atoms are around 0.0 nanometers. Okay. You don't need to know the exact size, but you should know the relative rank. You should also know that things smaller than a frog egg, you really need a microscope to see, with a light microscope, seeing down to the smallest prokaryotic cells. And then viruses are something that you can't see with a light microscope. You need an electron microscope to see them. Any question about any of that? Now, there is one error in the chart here. It has this point listed as 1 micrometer, and there's 1 micrometer here. This 1 micrometer is correct. This one should be a millimeter. And I'm not sure why the publisher never corrected that, because that's been repeated something like 12 times in the regular textbook, 3 times in the edition made for Clark College. So let's talk a little bit about molecules. We're going to talk about organic molecules and inorganic molecules. An organic compound is typically a complex structure, much more complex than an inorganic compound. And a compound is just a molecule. Organic compounds typically are held together by carbon. Carbon combined to 4 other atoms. And carbon typically holds most of the covalent bonds in an organic compound. An inorganic compound typically lacks carbon. They are required by living organisms for the organism's growth, repair, maintenance, and reproduction. And what are some examples of an inorganic compound? Come on, somebody guess. I mean, if you read the slide, you can find it there too. All right, you guys can give some examples of an inorganic compound. I am going to say that water is one of the most important inorganic compounds and is one of the most important to life, as well as the most abundant in living organisms. Let me shrink that a little. So let's talk a little bit more about water because it is so important to life. Water is a polar molecule. A polar molecule is one that has partial charges on the ends of it, and those charges can lead to the formation of hydrogen bonds with other polar molecules. So here's H2O, water. The oxygen doesn't share the electron equally with the hydrogens, and so oxygen has a partial negative charge. Hydrogen has a partial positive charge. This is not a complete charge, so it is not a +1. It is a partial charge. And unfortunately, the author of your textbook doesn't use the term partial charge. I'm trying to remember what term he uses, but it's something nondescript. It is not a complete charge. It is I think he said something like maybe a slight charge. I don't remember. Water, because it has a negative end and a positive end, will be attracted to other water molecules because the positive hydrogen will be electrically attracted to the negative oxygen. This creates a hydrogen bond between water molecules. And that hydrogen bond is what gives water its liquid properties, meaning the hydrogen bonds break and reform all the time, which is why liquid water can take the shape of any container you put it in, because liquid water will break its bonds and it will change its shape to fit the container it's in. And that's what makes water a liquid. The hydrogen bonds, which break and reform all the time, meaning this one can break and it can reform, or this one can, the negative oxygen can bind to another positive hydrogen like that one there. Like if this one breaks, that one can then form a hydrogen bond there. And that gives water the liquid properties. You should understand that water has two chemical bonds. It has hydrogen bonds between water molecules, and then it has polar covalent bonds within the water molecule between the hydrogen and the oxygen. That's a polar covalent bond. Any question about any of that? That's the hydrogen bond forming and breaking all the time, which we have in liquid water. There it's broken, formed with another molecule. All right, any questions about any of this? Well, here's a question for you. When does water never have hydrogen bonds?
When it's a vapor?
Yeah, when water is a vapor. So gaseous water does not form hydrogen bonds with other water molecules. And gaseous water is a gas, not a liquid. So it doesn't form hydrogen bonds. And then the other question is, when does the hydrogen bond between the water molecules never break? In which case water does not change its shape. When does water never change its shape? All right, I would have thought somebody would have known that. When water is a solid, we call it ice. The hydrogen bond forms and it does not break because the energy in the water molecules is not strong enough to break that hydrogen bond. And ice, as you know, does not change its shape because the hydrogen bond does not break and then reform. Water is a good solvent. All polar molecules and ions dissolve in water. So another polar molecule is one that has partial negative charges on one side and partial positive charges on another side of the molecule. So that's a polar molecule. An ion is a molecule that has either a +1 or a -1 or a multiple of a +1, meaning like +2. Sodium chloride is an ion, meaning the chloride ion is a -1 and the sodium ion is a +1. And water is very good at dissolving salt because the water comes up to, let's go to that one, comes up to one of the molecules, and then the water surrounds it. Let me blow this up, where the partial positive charges of water are attracted to the negative charge of chloride ion. And you have to have more than one water molecule surround the chloride ion to get it to go into solution. But water is a small molecule and chloride ion is a big molecule, or I should say atom. And so several water molecules can get around it. And then that will take this ion out of the crystal salt and then make it an ion in water surrounded by water, such as we have salt water. And the same is true with the sodium ion. The difference is sodium has a +1 charge. The oxygen will come around the sodium ion, and the negative partial charge of water will surround the +1 charge of sodium, and then the sodium ion will be pulled out of the crystal salt and be dissolved into the water like this. And like I said, that's how we get salt water. So ions can dissolve in water as well as polar molecules can dissolve in water. How do I want to show a polar molecule? What we could do for showing a polar molecule is, excuse me, still have my frog in my throat. Like if we had glucose here instead of this water molecule, glucose is a polar molecule, and then the negative side of water would be attracted to the positive side of glucose. And then the positive side of water would be attracted to the negative side of glucose. And that's why polar molecules can dissolve in water. Any question about any of that? So like I said, water is a good solvent because many molecules can dissolve in it. And all polar substances, meaning things that are either polar or ionic, can dissolve in water. Only nonpolar substances cannot dissolve in water. And an example of a nonpolar substance would be oil or butter. And as you know, when you make like salad dressing, homemade salad dressing, and you put vinegar and water in the salad dressing, and then you put the oil in and you shake it up, it'll separate from the water side and the oil side because the oil is nonpolar. It does not dissolve in the water. Any questions about any of that? So polar molecules are hydrophilic. Literally, that means water, hydrophilic lover. So water lover. But what we mean when we say that polar molecules are hydrophilic, we mean that polar molecules dissolve in water. Non-polar molecules are repelled by water, and we call them hydrophobic. Literally, this means fear of water, phobic, fear of water, and they do not dissolve in it. And like I said, oil or butter are hydrophobic substances, and that's because they're non-polar. Any question about any of that? Hydrogen bonding between water molecules makes water an excellent substance for buffering the temperature. You guys probably know that because in Western Washington and Western Oregon, probably Western California, the temperature is buffered by the Pacific Ocean. And what it is the water in our air tends to buffer the temperature. And that's why the temperature in the western parts of the states is milder and buffered compared to the eastern parts of the state. Like in eastern Washington, in the summertime, it's very, very hot. And in the wintertime, it's very, very cold. And in western Washington, the temperature is buffered. It's not as hot and it's not as cold. Any question about that? What happens is water is very good at absorbing heat, and it can absorb heat in two different ways. All molecules can absorb heat. Heat is just one form of energy. All molecules can absorb energy or heat, and then the molecules, once they've absorbed the energy, they will vibrate faster and move around much more. And that is actually a measure of the temperature. The temperature, all it is the measure of how much the molecules in the substance are moving around. And something that's hot has molecules moving around much greater than something who is cold because its molecules are not moving as rapidly. Like I said, temperature is just a measure of the movement of the molecules within the substance. And water can absorb energy into the molecule of water, and then the molecule will move around more greatly as the temperature increases. Water has a second way of absorbing the energy that most other substances do not have. And that is water can absorb some of that energy, some of the heat, and then that energy will be taken in by the hydrogen bond and eventually break that hydrogen bond. So some of that energy in water is absorbed by the molecule moving around, and some of it is absorbed by the hydrogen bond, which will break the hydrogen bond. As the hydrogen bonds are broken, the water molecules will break off and water will evaporate, so that the molecule of water that has absorbed the most energy will be the one that evaporates. And that tends to keep the water cool because the hottest water molecules are the ones that evaporate, and they take the heat away with it. Any question about any of that? This property makes water excellent at cooling things. And you should know that when you perspire, the water molecules absorb that energy and then evaporate, which tends to cool you because the hottest water molecules are the ones that evaporate and they take the most energy away. Any question about any of that? Now, something like oil, which does not form hydrogen bonds, can only absorb the energy by the molecules vibrating and moving around. Oil does not have hydrogen bonds. And so when you give the same amount of energy to oil as you give to water, the temperature of the oil will raise much greater than the temperature of the water. Any question about any of that? All right. Water can split into H plus and OH minus. On the surface of the Earth, water molecules normally only split into, meaning H2O, normally only split into H plus and OH minus on the temperature of the Earth. And that's assuming you're not putting electricity into the water molecule, encouraging electrolysis, which we're not going to talk about. Only on the surface of the sun does enough energy go into a water molecule to split all the hydrogens and the oxygen away from each other. Okay, but on the surface of the Earth, water only breaks into H plus and OH minus, and that doesn't happen very often, but it does happen. This causes charged molecules to occur in the water, and these charged molecules participate in chemical reactions, and the charged molecules of the water breaking apart give rise to the pH. Any question about any of that? All right. So remember, on the surface of the Earth, water does not break into H plus ions and O minus 2 ions. It only breaks into H plus and OH minus. The H plus is a proton. That's what hydrogen, the simplest form of hydrogen is. And then OH minus is a hydroxide ion. We will occasionally use these terms. Any question about any of that? All right, let's talk a little bit more about pH. When water breaks apart, forming H plus and OH minus, we have water at a pH of 7 because the concentration of the H plus ions and the OH minus ions equal each other. Any question about that? All right. So normally when water breaks apart, the first time it does, the H plus concentrations will be equal to the OH concentration. And when that happens, where am I? Here? No, here. When that happens, water is neutral. So pH of 7 is neutral. When we have more H plus ions in solution than OH minus, the pH of water drops. So urine is about a pH of 6, and it has more H plus in solution than OH minus. Wine and tomato juice are around pH 4, and they have much more H plus ions in solution than OH minus. Lemon juice is even more acidic, and it's pH 2, and then stomach acid is even more acidic than lemon juice, and it's around pH 1.5. And this really depends on what food you have in your stomach and how much gastric juices the stomach is producing. But normally stomach acid is around pH 1. The only thing that's more acidic than stomach acid would be the carbaddiary acid. And I think that's around pH 1, maybe a little bit less than pH 1. Okay. Any question about any of that? Seawater is a little above pH 8, and seawater has more OH minus in solution than H plus in solution. Milk of magnesia is even more basic, and its pH is above pH 10, around pH 10.4 or 5. And it has much more OH minus than H plus. Let's see now. Household bleach, even more basic, pH above pH 12, much more OH minus than H plus. And then probably the ingredient that you're most familiar with, oven cleaner, is even more basic. And I doubt most of you are familiar with lime water. That's even more basic. Lime water is very close to pH 14. And so lime water would have the most OH minus in solution compared to H plus. All right, any question about that? Anything that is above pH 7 is alkaline or basic. Alkaline and basic are the same. There's more OH minus in solution than H plus in solution. And then anything that's below pH 7 is acidic, and it has more H plus in solution than OH minus in solution. Neutral has to have exactly the same H plus and OH minus, so only pH 7 is neutral. And if you have pure water, it will be pH 7. If you don't know, carbon dioxide will dissolve in water, and it will lower the pH of water down to around pH 5 or 5.5, something like that. Let me go ahead and show you why. So you have H2O, and it breaks apart into H plus and OH minus. I'm not going to superscript the charges there. All right. And right there, that's pH 7. When carbon dioxide, CO2, plus water dissolves in water. Oops. The carbon dioxide, oops, and form carbonic acid. Oop. And this carbonic acid can break apart into H plus, which is acidic, and carbonate. Oops. With a negative charge. Anyways, that's why distilled water will lower its pH, and this pH it's someplace around 5.5 to 5. I'm just going to put pH equal to around 5. That's not an equal, is it? Can you guys see this? I'll try and blow that up. Oh, that's not right. All right, let's talk a little bit about what the pH means. pH means something. pH is the measure of H plus in water solution. pH of 7 equals 1 times 10 no, that's not right. That's 1. It's 1 H plus ion for every 1 times 10 to the 7th water molecules. And I'll put that superscript to make that clear. Can't do it there. Everybody clear on that? A pH of 7 means there's 1 hydrogen ion for every 1 times 10 to the 7th water molecules. What does a pH of 6 mean? We're going to go 6.0 equals 1 hydrogen ion. Anyone know? Anyone want to guess? Nobody wants to guess? It's 1 times 10 to the 6 water molecules. And then a pH of 5 is the same. Let me get all of that. So a pH of 5. 1 times 10 to the 5th. pH of 8. What's your guess? What number did I put in here? Come on.
8.
Yeah, good job. Yeah, a pH of 8 is 1 hydrogen ion for every 1 times 10 to the 8th water molecules. And that's what the pH stands for. So the pH does mean something. It's the measure of the hydrogen ions in water solution. All right. Let's talk a little bit about acid, bases, and salts. An acid is a substance that gives up 1 or more H plus ions in solution, like hydrogen chloride is hydrochloric acid, and it's an acid because when you put hydrogen chloride in water, the H plus will dissociate, releasing more H plus in solution. And that's why HCl is an acid. A base, the textbook says, is a substance that dissociates, giving up 1 or more OH minus, meaning hydroxide ions. Like sodium hydroxide is a base because it will, when dissolved in water, break into sodium ion and then hydroxide ion. And the hydroxide ion being in solution will increase the number of these in solution. And that's why sodium hydroxide is a base. The book does not tell you, and I don't know why it does not, but a base is also a substance that can absorb hydrogen ions, like bicarbonate, such as in sodium bicarbonate, can absorb H plus in water, forming carbonic acid, and that will absorb and reduce the hydrogen ions in solution. And that's why bicarbonate or sodium bicarbonate is a base. All right, so remember with a base, it's a substance that either gives up OH minus in solution or a substance that absorbs H plus in solution. Any question about any of them? All right. So that is an acid and that is a base. A salt, on the other hand, is a substance that dissociates into an ion, well, into ions, a cation, a positive ion, and a negative ion, an anion. However, neither of these ions can be H plus or OH minus. So table salt, sodium chloride, is a salt because it dissociates into a cation, sodium ion, and an anion, chloride ion. However, by this definition of a salt, magnesium chloride is also a salt because it'll dissociate into magnesium plus and 2 chloride minuses. And so magnesium chloride is a salt. Normally, we do not call, in common usage, we only call sodium chloride a salt. But in chemistry, any substance that dissociates into a cation and an anion is a salt. Salts are electrolytes. An electrolyte is a substance that carries an electric charge in solution. So salt water is an electrolyte because sodium and chloride can carry the electric charge in water much better than water can carry an electric charge. And that is why during a lightning storm, you should not be in the ocean water. Because if the lightning bolt hits the water any place near you, it will transfer the electric charge to you and you will receive an electric shock. Okay? And I'm talking about anywhere near you. This could be, I don't know how far away, but much further away than if you were in a freshwater body of water. Okay? So you should get out of the ocean during a lightning storm. And if you're in a freshwater body and there's a lightning storm, if the lightning bolt hits within a few feet of you, you will get an electric shock. It wouldn't be as great as if we were in salt water, but you would get an electric shock. And if it's within a few feet, that could do damage to you, could even kill you. Any question about that? So electrolytes are salts dissolved in solution, and sodium chloride is both a salt and an electrolyte, and that's why salt water carries an electric charge much greater than pure water. All right, organisms need to maintain a fairly constant balance of cellular acids and bases. We have a term for that. You don't need to know it for microbiology, but you probably had it in your previous biology class, and that's called homeostasis. All organisms control the pH within the organism, and how they do that is by using buffers. The main buffer in our body, there are other buffers, but the main buffer in our body is actually sodium bicarbonate, which can separate into this. Let's go ahead and remove that. And then if needed, this is unlikely to happen in our blood, but if needed, that can go further so that HCO3 can break down into H plus. Oops. Oh, I got the wrong one. That's why I'm not getting it. And then CO3. And that's going to be a -2. Oh, fudge. Not doing too good typing here. And let's make that superscript. Like I said, HCO3 in the blood is unlikely to break into H plus and CO3 -2 because it would be you would have to require such a basic pH that for this to happen, that would kill you at that extreme pH. Anyways, that's the main buffer in the blood, and let's go ahead and take this apart. And the reason why this is a buffer is that this reaction can go the other way. So let's just reverse the arrow here. Actually, I don't need to get there. I need to go just here. Oops, I took care of it. Darn. Oop. There we go. I'm just going to copy and paste that. So H plus and CO3 -2 can form bicarbonate, and then HCO3 -1 plus H plus can then form carbonic acid. And this is why this is a buffer, and that is that these reaction, actually there is, I guess I can put that up there too. And let's not do that. We can put this up here. If you get too much acid in your blood, what happens is, I'll get it eventually, you breathe out CO2. And that's why if you've ever seen some kids chugging some Coke, why the Coke, which is acidic, doesn't kill them, is that they have buffers in their blood. And if need be, the bicarbonate in their blood forms carbonic acid. And if need be, they can simply breathe out the CO2 to remove this excess acid from the Coke. Okay? And that's why kids who chug Coke don't die, and that is they have excellent buffers in their body. Let me think about that. If you were to need more acid, you could take CO2 and water in your lungs to form bicarbonate, and then that could form H plus in the blood if your blood did need more acid. So I guess that's another way that could happen too, is you can breathe in CO2 and form acid in your blood if needed. I didn't ever think of that until now, but I knew that you can breathe out the carbonic acid. Any question about any of that? Those are the main buffers in your blood. We do have other buffers, but this is a very simple one that is simply involving carbon dioxide and water. And it is one of the main buffers in the body. All right, I've already talked about pH. The pH, I haven't mentioned this. The scale of the pH runs from pH 0 to pH 14. At pH 0, let's go to my thing if I still have it. Yeah, I do. At pH 0, no, I don't. Yes, I do. It's right here. At pH 0, Yep. You have one hydrogen ion. For every 1 times 10 to the 0. Oops. Oh, good. I still have that. Up. 0 water molecules. And if you don't know, 1 times 10 to the 0 is equal to 1, meaning that every water molecule would then break into H plus and OH minus. Okay, so you have 1 H plus for every water molecule. And you can't go below, obviously, a pH of 0 because the water molecule breaks into H plus at pH 0. Any question about that? That's the pH scale. 0 is the lowest, 14 is the highest, and I don't know why the pH scale doesn't go above pH 14. Somebody can research that if you want, but most organisms grow best around pH 7. So something like pH 6 to 8 is the pH that most organisms grow best in. And if you try to grow most bacteria in something that's less than pH 6, the bacteria, most species, would not grow because it's just too acidic for them to grow. Any question about any of that? And the same thing, most organisms will stop growing after about pH 8 because it's too basic for the organism to grow. I'm talking about bacteria. Now, there are exceptions to that, but I'm talking about, generally speaking, most bacteria or most organisms grow best between pH 6 and 8 or someplace around pH 7. All right, any questions about anything we've discussed? All right, let's talk a little bit more about organic compounds. You're seeing several organic compounds here. And generally, organic compounds are fairly complex, like this one here, or these here. This is the simplest organic compound, acetic acid. Well, I guess methane's the simplest. So these are simple ones. The main point is that carbon tends to form most of the covalent bonds linking the molecule together. So you look any place here, it's linked together by carbon. It is true, like right there, it's linked together by oxygen. Right there, also by oxygen. But most of the links are by carbon. Not all of them, but most of them. Another thing you'll note is that organic compounds always contain carbon and they always contain hydrogen. So the simplest organic compounds are simply called hydrocarbons, and they only contain carbon and hydrogen. And all of these are hydrocarbons. Okay, I think all of these are hydrocarbons, but I'd have to look carefully, blow that up to make certain that there aren't other. Yeah, there's no other elements in there. So those are hydrocarbons as well. So hydrocarbons are organic compounds, and hydrocarbons only contain carbon and hydrogen. And like propane and ethane are gases which burn, and fossil fuels tend to be hydrocarbons. You do not need to memorize any of these elements. I'm just trying to show you what some hydrocarbons look like. So organic compounds always contain carbon and hydrogen, and they also tend to contain Oxygen. Not all organic compounds contain oxygen, but many of them do. Organic compounds may contain other elements, such as nitrogen, sulfur, and phosphorus. And all of these elements are held together by covalent bonds, usually by carbon, a carbon bond binding to carbon. Compared to inorganic molecules, organic compounds are more complex and they're capable of more complicated biological functions. I've already showed you some organic compounds, but can anyone name some important biological organic compounds? Anybody? All right. The four biological molecules, proteins, carbohydrates, lipids, and nucleic acids are all organic compounds. And then all of the derivatives like glucose, starch, are all organic compounds. Carbon can bond with up to 4 other atoms. So this carbon here can bind 1, 2, 3, and then to the OH group, meaning to the O, and it can bind 4 times. Carbon atoms often bond together in chains or ring structures, and I showed you one right here. These are chain or ring structures. Actually, that's a chain structure, sorry. These are ring structures. So the carbon skeleton is the backbone of the organic compound, and the backbone is simply where carbon is bonding to carbon in an organic molecule. Like in ethanol, this right there is the backbone. In isopropanol, that right there is the backbone. Organic compounds can have functional groups that bind to a carbon, and the functional group gives the molecule different properties, different behaviors than the molecule without that functional group. Like methane is a molecule that does not dissolve in water. Methanol, which has the functional group OH, can dissolve in water. Methane is a gas and methanol is a liquid, at least at standard room temperature and pressure. Any question about that? So organic compounds tend to have functional groups binding to them, and the functional group tends to give the molecule important properties. Most of the chemical and physical properties of organic compounds come about because of the functional groups. OH, the hydroxyl group in alcohols, is a functional group, and the OH gives the molecule the ability to dissolve in water and be attracted to water. The functional groups shown in a red box are the functional groups that you need to know. So you need to know the OH functional group, the NH2 amino functional group, the carboxyl group, the COOH group, and that also sometimes said COOH, and then the phosphate group, the PO4 group. And the phosphate group has a negative charge on it. Why do you think you need to know these functional groups? Because you need to study these molecules, and their molecules we're going to be talking about, and functional groups we're going to be talking about. So the OH group is actually in many of the biological molecules, such as the lipids, the carbohydrates, and the OH, it's not listed here, is also in nucleic acids and in proteins. So the alcohols and all the biological molecules. The NH2 group, the amino group, is in proteins. The carboxyl group is also in proteins, and it's in lipids. If you know what an organic acid is, the carboxyl group is in organic acids also. And then the phosphate group is in nucleic acids such as DNA and RNA, as well as in ATP. And we'll talk about ATP in a little bit. All right, when we're talking about the organic compounds, we have different sizes of them. And for many of the organic compounds, actually most of the organic, not the most of the organic compounds, most of the 4 biological molecules, if it's a large molecule, it will be made up of smaller molecules. So small organic molecules can combine into the larger macromolecules. The macromolecules we call polymers, and they consist of many small molecules called monomers. For example, I think I got that right here. Well, no, I don't have it right here. I'll give you the example in a little minute. Oh, I'll give you the example now. Glucose, for example, could be monomers, and you put many of the glucose molecules together, that's many monomers put together, you can form a complex polysaccharide or a complex carbohydrate that we call starch. All right, the monomers shown here, the R stands for anything, the OH group of one part of the monomer can combine with the H part of another monomer, and the R can stand for anything else. Anyways, these are 2 monomers, and the water, the OH of this group and the H of that group, can come off, forming water, and then this R here will be directly covalently bonded to this R group here, forming RR, and that is a condensation reaction, and it's also called a dehydration synthesis because water is pulled out here and formed here, and that's why it's called dehydration synthesis. You can do the reverse, add water to a polymer, meaning RR, and that water will split the R and break apart with the H going on one R and the OH group going on the other R. So the RR breaks into ROH and then R, and the water, of course, is broken apart there. And then we get the monomers back. This reaction going this way, adding water to a polymer to form the monomers, is called hydrolysis, using water to split the molecule. So dehydration synthesis is a mechanism for making large macromolecules such as carbohydrates, lipids, and proteins. And then hydrolysis is a process for making a larger molecule and then breaking it into smaller molecules, such as taking a polymer and then breaking it into the monomers. Any question about any of that? You should be familiar with hydrolysis because Marty McFly in Back to the Future explained it when his mother took this hockey puck and put it into her pizza machine, and then lo and behold, by hydrolysis, out came a pizza, and Marty McFly said, "Ma, you sure know how to hydrate a pizza." You remember that? That was hydrolysis. This is showing you just in the biological form, dehydration synthesis, where we take a monomer and the OH group comes off the monomer and the H group comes off of the polymer actually there to add that monomer onto the growing polymer. And then water comes off. And then hydrolysis is you add water to the polymer, and then the water splits apart with the H going to the smaller polymer and the OH being added to the monomer. So hydrolysis and condensation are simply the reverse of each other. All right, let's begin talking about the 4 biological molecules. The first one we're going to talk about is the carbohydrates, and these include sugars, starches, and other complex carbohydrates. Carbohydrates are important as an energy source, so we need to eat them to get energy, but they're also important for the structure in our cells and in the makeup of nucleic acids. Although a carbohydrate is not a nucleic acid, when we're building a nucleic acid, we can begin with the carbohydrate. Carbohydrates consist of carbon, hydrogen, and oxygen, but not in any ratio. Carbohydrates tend to be in this general formula. For every 1 carbon, there are 2 hydrogens and 1 oxygen. And so for every carbohydrate, we can simply go CH2O in parentheses and then put N for the carbohydrate. Like for glucose, this would be CH2O in parentheses and then 6. So glucose is C6H12O6. Any question about that? Carbohydrates include things as simple sugars or strings of sugars linked together. We can classify carbohydrates into 3 major groups based upon their size. There's the monosaccharides, the smallest carbohydrate, or the simple sugar, the disaccharide, which are simply 2 monosaccharides put together, and lactose is an example of a disaccharide. Let me see, lactose is beta galactose hooked together to glucose, meaning making up lactose. And then a polysaccharide, which is many monosaccharides linked together to form the polysaccharide. And starch would be an example of a polysaccharide, where all starch is a bunch of glucose held one after the other to make the polysaccharide. So monosaccharides are the simple sugars. All simple sugars or all monosaccharides contain 3 to 7 carbon atoms, but usually a monosaccharide has either 5 or 6 carbons, like glucose and fructose and galactose all contain 6 carbons. If you are familiar with ribose and deoxyribose, I think those are the only sugars we'll ever talk about that have 5 carbons. But most sugars have 5 or 6 carbons. Monosaccharides are sweet. They're water-soluble molecules. Disaccharides are formed by joining 2 monosaccharides in dehydration synthesis. So we can take glucose, C6H12O6, plus fructose, also C6H12O6, pull out the water in the glucose and the fructose in dehydration synthesis to get sucrose, which is C12H22. I can't read that, sorry. C12H22O11. And then we have water over here. And we can break sucrose down by adding water, which will then break apart and part going here and part going there. And that's hydrolysis, so that sucrose can make fructose and glucose, two monosaccharides. Carbohydrates or monosaccharides, the carbon atoms may be in a line. Oh, I don't have that shown here, but hopefully you know what I'm talking about, in a linear structure. Or they may be in a ring, like this glucose right here is in the ring form. Let me show you the line form. Let me see what slide I'm on. I'm on slide 22. Not seeing it. No, that won't be good. This will be good enough. That's showing you a linear structure of the molecule. This is not the molecule we're looking at. This is isopropanol, which is not a sugar, but it's showing you there we go. There it is there. There's a linear form. And then that's a ring form. 22. If you don't know, fructose is usually in the circular form, at least at standard temperature and pressure and normal pH. And then glucose is usually in a linear form, at least at standard temperature and pH. Carbohydrates do include the polysaccharides. The polysaccharides consist of many monosaccharides. The polysaccharides can be straight or branch chains. The simplest example of a polysaccharide is starch, shown here, which it's actually much longer than this. It's just showing you the basic shape. And that's a glucose, 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 that's a glucose. So starch, all it is a bunch of glucose molecules hooked together one after the other in mostly a linear straight chain. Okay. And here are several examples of starchy foods: bread, cereals, pasta. You probably didn't know beans and chestnuts have a lot of starch in them. About a third of the weight of beans and chestnuts are starch. Okay. And it is true that beans, in particular, chestnuts also, do have a lot of proteins. And actually only about a third of the weight is protein. And then the other third, at least of beans, I'm not sure about chestnuts, but about a third of the weight of beans is lipid. So beans would be about a third, and a third. But it is a starchy food, as well as a protein food and a lipid food, I guess. Some other examples of carbohydrates are glycogen. That is the way that animals store glucose in our liver and our muscle cells. We store glucose as glycogen. Plants tend to store glucose as starch, and that's why we get cornstarch and potato starch from corn and potatoes, because plants tend to store glucose as starch. There are some examples like cane sugar and sugar beets. They don't store glucose as starch. They tend to store it as sugar, meaning as glucose? No, as sucrose. They tend to store it as sucrose. Cellulose, another example of a polysaccharide. Chitin, we're not going to talk about. let's just briefly look at this slide. You don't need to memorize any of this. It's just showing you starch, mainly a straight chain of glucose, one after the other. Glycogen is similar. All it is starch, glucose held one after the other, but glycogen branches much more often than starch. So glycogen is a branched glucose chain. Cellulose is also a molecule only made up of glucose. Let me see if I can blow this up so we can see that better. So here is the chain of glucose, one after the other. And here's another chain of glucose, one after the other. You see these broken lines? Those are not covalent bonds. These are covalently held together. These are hydrogen bonds forming between the glucose. And if you notice, there's a lot of hydrogen bonds in cellulose. And that is why when plants make cellulose in their cells, many animals like grass cannot digest it. That cellulose is difficult to digest because of all of the hydrogen bonds as well as the covalent bonds. And not many animals can digest it. Cows, for example, can, but the cow itself doesn't digest the cellulose. The cow has in his stomach a microbe which actually digests the cellulose. And then once the microbe has digested the cellulose, the microbe gives some of the digested cellulose to the cow, and then the cow can digest the simpler, meaning this, the cow could probably digest this chain of glucose, which would be very similar to starch. And I'm sure the cow can digest starch, as well as if the microbe digests it into glucose, the microbe could give the glucose, some of the glucose to the cow, and then the cow can digest glucose. So the cow itself does not digest cellulose. It has a microbe living in one of its four stomachs that digests the cellulose. Chitin, another molecule we're not going to talk about. I'm going to briefly show you peptidoglycan. We'll look at this greater in a, oops, didn't mean to do that. We'll look at peptidoglycan later in this class, but peptidoglycan is at least mostly carbohydrate. It's hooked together by two sugars linked together, one after the other covalently, and then there's a chain of them. The chains repeat, and then the chains can be linked together by a short peptide. And we'll talk more about that later. But the point is that peptidoglycan is at least mostly a carbohydrate. All right, any question about any of that? All right, I think I'm going to stop here. Well, let me just briefly state this one, and that is lipids consist of carbon, hydrogen, and oxygen. They do contain oxygen, but they do not contain much oxygen. Lipids are non-polar molecules and insoluble in water, meaning they're hydrophobic. We use them for structural and energy storage roles. Like, for example, if you eat too much food, you will store the excess down as lipid, a fat in your body. And then we do use lipids as a component to build cell membranes and other parts of cells. Lipids can be broken into three types: simple lipids, complex lipids, and steroids. Let me do state that most lipids are nonpolar and insoluble in water. There is one exception. There is one lipid which is soluble in water, and we'll talk about that more next time. Any questions about any of that? All right, I think, let me check the time here. I think we're done with the lecture, but just to make sure, yep, we're done with the lecture. I'll see you at 6:30 for the lab if there are no questions. All right, bye.